When you watch movies like Star Wars or Lord of the Rings, there's always this big deal about destiny and bloodlines passing down something pure.
Right, the legacy, like Luke Skywalker needing to be the good guy because of his father, or Aragorn and his lineage.
Exactly. But what if the actual biological process of creating that legacy,
Yeah.
well, what if it changes dramatically the older you get?
Now you're hitting on exactly what we're digging into today.
Yeah.
We're exploring some pretty hidden age-related genetic risks tied to fatherhood.
Okay.
We're going deep, like down to the molecular level, to understand how aging in men kicks off this almost selfish competition among the stem cells that actually produce sperm.
Selfish competition. Okay, that sounds intriguing. So this deep dive, what's our source material?
We're looking at some really cutting-edge work here. It's a paper titled "Sperm Sequencing Reveals Extensive Positive Selection in the Male Germline," published in Nature, which is, a top journal. And the lead authors are Matthew D.C. Neville and Raheli Rabari, working with a team mostly from the Wellcome Sanger Institute over in the UK. Really solid research.
Got it. So our mission today is basically to understand this cellular competition, this selfish thing, and how it bumps up the risk for certain developmental issues in kids when dads are older.
That's the plan, to really break down the how and how much.
All right, let's start with the basics. The genetic clock, I guess. We know sperm are produced throughout a man's life, right? From these spermatogonial stem cells.
Exactly. They're constantly dividing, and every single time a cell divides, there's a tiny chance of a mistake, a mutation creeping in.
And a study actually put a number on that.
They did, and this is pretty amazing. They found a really consistent linear rate. It accumulates at 1.67 mutations per year.
Okay, 1.67 per year.
Per year, and that's in the haploid genome, basically, the genetic info that one sperm cell carries. So the clock is ticking, predictably adding a bit more risk each year.
So a 50-year-old father is starting with a different genetic slate in his sperm compared to when he was 20, just from this baseline accumulation. Do we know what kind of mistakes these are?
Yeah, they trace them. These mutations mostly match two specific patterns or signatures that scientists already link to aging. They're called SBS1 and SBS5.
SBS1 and SBS5.
Okay. Think of them as signs of just the normal wear and tear that happens when DNA gets copied over and over again for decades.
Now, here's something interesting they found, kind of counterintuitive. They compared sperm to blood cells from the same men. Right, and get this. Blood cells accumulated mutations way faster, about 7.6 times faster than the sperm stem cells. 7.6. Wow. So the cells making sperm are actually quite protected.
That's the key takeaway there. The male germline, it's like a VIP section, relatively shielded from mutations compared to other parts of the body. But critically, even in this protected zone, that 1.67 per year clock is still ticking away reliably.
Okay, but measuring just 1.67 mutations a year sounds incredibly difficult. Standard genetic sequencing usually has errors, right?
Yeah.
How could they be so precise?
Oh, that was the technological hurdle. Standard methods are just too noisy for this. They'd introduce their own errors, masking the real low-level mutations.
So what do they use?
They use this super high-fidelity method called nanosec. It's a type of error-corrected duplex sequencing.
Duplex, meaning?
Basically, instead of reading a DNA strand just once, it reads both strands of the DNA double helix, then it compares them. This lets them cancel out most of the random sequencing errors. It's like checking your work really, really carefully.
Okay, so it gives you much higher confidence in the mutations you do find, even rare ones.
Exactly. It gives you single molecule resolution. That's how they could analyze sperm from 81 guys, aged all the way from 24 up to 75, and get this clear picture of mutation accumulation.
All right, so we have mutations building up slowly but surely. Now let's get back to that selfish cell idea. That sounds like where the real action is.
Yeah, this is the core story. It's called positive selection. The issue isn't just that random mutations happen, it's what happens when one of those mutations gives the stem cell itself a competitive edge.
Okay, break that down. What does a competitive edge mean for a sperm stem cell?
Well, imagine you've got loads of these stem cells lining the tubes in the testes, all making sperm. Now, one stem cell gets a particular mutation. Let's say this mutation makes it divide just a little bit faster or survive a little bit better than its neighbors.
Okay.
That cell starts to multiply more. Its descendants, its clone, start taking over more space in the tube, producing more and more of the sperm. It's selfish because the mutation benefits the stem cell's survival and replication, not necessarily the health of the potential offspring.
Ah, I see. So it's like a rogue employee finding a shortcut to boost their own output, and they start dominating the factory floor, pushing out the regular workers.
That's a great analogy, and the research shows this whole process, this internal competition, actually gets stronger as men get older.
How do they measure that?
They used a metric called the DNDS ratio. It's basically a way to measure how much positive selection is happening across all the genes. A ratio around 1 means things are pretty much random, no strong selection.
But what did they find age-wise?
In the younger guys, aged 26 to 42, the ratio was 1.01. So basically negligible selection, just random mutations ticking along.
Okay.
But in the older group, 59 to 74 years old, that ratio jumped up to 1.09.
1.09, so definitely not random anymore. Selection is actively favoring certain mutations in older men.
Exactly. The older you are, the more intensely these selfish clones are competing and expanding.
Now, I remember reading that earlier ideas about this focused on mutations that sort of turbocharge a gene, turn it on aggressively. Did this study find that was the main mechanism?
Actually, no. That was one of the big surprises here. While they did confirm some of those known activating mutations are involved, especially in pathways like the RAS-MAPK pathway.
Okay. Their more sensitive nanosec method found 40 genes in total, showing signs of this positive selection. And get this, 31 of those were completely new finds. 31 new genes being selected for in sperm stem cells. Wow, what kind of mutations did they have?
This is the twist. For 30 out of those 31 new genes, the advantage came from loss-of-function mutations.
Loss of function. So mutations that break the gene or turn it off. How does that give a cell an advantage? It seems counterintuitive, right? But think about it. Sometimes the gene that gets turned off might be one that normally puts the brakes on cell division or triggers cell death if something goes wrong.
Ah.
So if you disable the brakes,
You go faster. The stem cell can divide more recklessly, ignore signals telling it to slow down or die. It gains a competitive edge by essentially disabling some of its own safety mechanisms.
so the ways these cells get selfish are much more varied than we thought. It's not just about hitting the gas. Sometimes it's about cutting the brake lines.
Exactly. Activating and inactivating mutations can both drive this clonal expansion.
Okay, so mutations accumulate steadily with age, and then this selection process amplifies certain mutations, both on and off types, giving some stem cells an advantage. What does this mean for the resulting sperm and potentially a child? Are these selfish genes linked to diseases?
Yes, and that's the major concern. The genes that gain this advantage inside the testes are very often the exact same genes known to be involved in major developmental disorders in children,
Oh, wow.
and also genes linked to cancer predisposition. We're talking about really fundamental pathways that control growth and development, like the RAS-MAPK pathway we mentioned, but also WNT signaling, TGF-beta signaling. These are major players.
So the very thing that gives the stem cell a growth advantage of the testes, faster division, less regulation, is what can cause problems if that mutation is passed on to an embryo, leading to unregulated growth or development issues.
Precisely. It's a double-edged sword. Good for the stem cell clone, potentially bad for the offspring. They replicated some known culprits like SM84, but also highlighted several newly identified genes that carry a big part of the burden.
Like which ones?
Genes like NF1, CUL3, KDM5B, MIB1, and PTPN11. They calculated that just these 5 plus one other accounted for over 20% of the total disease or selection effect they measured across all the sperm samples.
20% from just a handful of genes. Okay, so let's get to the bottom line for someone listening. How much does this whole process actually increase the risk? Can we quantify it?
Yes, they put numbers on it. Overall, this positive selection process drives roughly a 2 to 3-fold increased risk of these known disease-causing mutations being present in sperm across all the ages they studied.
2 to 3 times higher because of this selfish selection.
Right. But the most striking number, I think, is for older men. They estimated that for men over 50, somewhere between 3 and 5% of their sperm carry one of these likely disease-causing mutations across the entire exome.
3 to 5%. That sounds significant. Is that higher than we thought before?
Substantially higher, yes.
Yep.
Previous estimates were generally lower. This nanosec data and the understanding of the selection process really bump up the potential risk associated with paternal age.
It really solidifies that paternal age is a significant factor in reproductive health, just like we've known about maternal age for things like chromosome issues.
Absolutely.
One last thing from the study, did they look at lifestyle? Like, does smoking or drinking or BMI affect this mutation rate in sperm?
They did look at that, and interestingly, using their statistical models, they found no significant link. BMI, smoking history, alcohol consumption, none of these showed a clear effect on the mutation rate in sperm or the expansion of these selfish clones.
Really? Because those things definitely affect other tissues,
And
right?
absolutely. They saw effects in the blood samples from the same men. So this finding really strengthens that idea we talked about earlier. The male germline seems to be this remarkably protected niche.
So it really boils down to age. Age is the main factor driving this specific type of genetic risk accumulation in sperm.
That's what this data strongly suggests. Age is the primary driver here.
Okay, this has been fascinating and maybe a little alarming. Let's try to sum up the main takeaways. What are the key messages here?
I think number one is simply that age is the crucial factor. Mutations just steadily build up in sperm stem cells, about 1.67 per year, like clockwork. And it's this aging process that sets the stage for everything else.
Okay, age drives the baseline accumulation. What's second?
Second is that selection dramatically amps things up. It's not just random accumulation. Specific mutations, and importantly, not just activating ones, but also loss of function ones, give certain stem cells a powerful competitive edge. This selfish selection makes disease-linked mutations much more common in the sperm pool than they would be otherwise.
Right. Selection makes the problem worse. And the third takeaway.
The third is the risk is real and quantifiable. Thanks to the selection process, the estimates for risk are higher now. Particularly for middle-aged and older men, maybe 3 to 5% of sperm could carry a mutation linked to developmental disorders. It's a significant number.
Age accumulates mutations, selection amplifies the risky ones, leading to a measurable 3–5% risk in older men.
Got it.
It definitely provides a clearer picture of paternal age risks.
Yeah, and it puts it more on par with the known risks associated with maternal age, although the mechanisms are totally different, obviously.
But the connection you mentioned earlier between these selfish genes in the testes and cancer pathways elsewhere in the body, that seems really important too.
It really is. It's kind of profound, actually. The exact same genes and the exact same mechanisms, like gaining a growth advantage by mutation that drive this selfish competition among sperm stem cells, are often the very first steps in how cancer starts in other tissues.
So it's like the testes are a sort of contained battleground for these potentially cancerous processes. You could almost think of it that way. This process is happening for decades, but it's mostly contained within the seminiferous tubules. So here's the really big provocative thought this research leaves us with. Okay.
If we could fully understand how the testes manage to contain this cellular competition, how they regulate these potentially rogue stem cells for so long, could that give us clues? Could it offer like an evolutionary blueprint for figuring out how to control or maybe even slow down that same kind of cellular selfishness that drives aging and cancer in the rest of our bodies?
Wow. So studying sperm production might hold keys to fighting cancer and aging more broadly. That's definitely something to think about.